Okay, well, thank you, Gary, for the introduction. I'd also like to thank Devor for the work he's put into this. It's quite a phenomenal amount of work, and I also like to thank the ISHR for providing me a platform to speak with you today. And as Gary mentioned, I'm going to talk about free fatty acid receptor 4 signaling in the heart. And since I know many of you are probably not familiar with the free fatty acid receptors, I thought I'd give a little background information. So this is a family of G-protein coupled receptors that bind to and are activated by endogenous fatty acids. And they were deorphanized roughly 15 years ago, and there are currently 4 family members, FFAR1, 2, 3, and 4. Now, FFAR2 and FFAR3, formerly known as GPR43 and GPR41, are receptors for short-chain fatty acids, carbon chains of 2 to 6. They're primarily expressed in the intestines, but FFAR2 is also found in adipose tissue, and they regulate intestinal homeostasis and immune responses. Conversely, FFAR1 and FFAR4, formerly known as GPR40 and GPR120, are receptors for medium to long-chain fatty acids, carbon chains of 10 to 24, which would include omega-3 and omega-6 polyunsaturated fatty acids that many of you are familiar with. And most of the work to date has focused on their role in regulating metabolic and inflammatory responses. So let's talk a little more specifically about FFAR4. Again, it's formerly known as GPR120. You see that in the literature quite often. And most of the literature suggests this receptor functions as a nutrient sensor for medium and long-chain fatty acids to regulate metabolism and attenuate inflammation. Now, pharmacologically, it binds to medium and long-chain fatty acids, carbon chains of 10 to 24, and that includes saturated fatty acids, monounsaturated fatty acids, and polyunsaturated fatty acids, and they bind the receptor with affinities in the low micromolar range. In terms of efficacy, polyunsaturated fatty acids are full agonists, where saturated fatty acids are generally considered at best to be partial agonists. FFAR4 is highly expressed in many tissues, including the lung and the brain, although its function there is still unknown. It's also found in many immune cells, including macrophages, as well as GI enteroendocrine cells like K and L cells, in pancreas, in alpha, beta, and delta cells, although its expression in beta cells is debated, and it's found in adipose tissue. In terms of signaling, FFAR4 is a GQ-coupled receptor, and it signals through G-alpha-Q to activate phospholipase C-beta, increasing intracellular inositol phosphates and releasing calcium, as well as activating PKC. But it's also been shown that the receptor can signal through arrestin-mediated pathways, specifically through beta-arrestin-2. And Jarolevsky's group has suggested that this could be cell type specific. For instance, they showed that in adipocytes, FFAR4 activates GQ to increase GLUT4 expression on the surface of the cell, whereas in macrophages, FFAR4 signals through arrestin-mediated pathways to induce an anti-inflammatory or M2-like phenotype. In humans, we have 2 isoforms of FFAR4, a short and a long isoform, but the short is the predominant isoform. The 2 isoforms are different by 16 amino acid insertion in the 3rd intracellular loop of the long isoform, rendering it incapable of signaling through G-alpha-Q. You can only signal through arrestin-mediated pathways. And all other species express only one form that is homologous to the short isoform, or the predominant isoform in humans. So how did we become interested in FFAR4 in the heart? And what is our rationale for doing this study? So it's based on our long-term interest in omega-3 mediated cardioprotection in heart failure. And all these studies that I'm going to show you today were done in collaboration with Greg Shearer's lab at Penn State. And we've been working on a paradigm where we use dietary supplementation with omega-3 using both EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid, which are the 2 cardioprotective long-chain omega-3s. And then we've looked at them in a model of omega-3, aortic constriction, excuse me. So when we give a dietary supplementation with omega-3s, then we perform aortic constriction, and we look to see what the effects of omega-3 dietary supplementation are. And we found that it prevents contractile dysfunction. You can see that here. Here's systolic dysfunction declined after TAC, diastolic dysfunction that's prevented by omega-3 supplementation. Furthermore, we've seen that omega-3 supplementation prevents interstitial fibrosis. So here's a traditional interstitial fibrosis response following TAC that's prevented by omega-3 dietary supplementation. And when we began to look at which specific omega-3s may mediate this effect, we found that EPA alone was sufficient to prevent this fibrotic response. DHA had no effect, but it turns out that DHA actually augments the effects of the EPA. In other words, it makes EPA more efficacious. And when we began to think about Well, what is the mechanism responsible for this omega-3 cardioprotection? The first thing we actually examined was omega-3 incorporation into cellular membranes in the heart, because at the time, this was the accepted mechanism of action. And when we measured this, we found that with dietary supplementation, DHA is readily incorporated into cardiac myocytes and fibroblasts in vivo, but EPA is not. So if we give EPA diet, we can see EPA levels are increased in red blood cells, but not in myocytes or not in non-myocytes. So if EPA is having a cardioprotective effect, it's doing so independent of membrane incorporation. So how do omega-3s protect the heart? Well, at this point, there's really 4 potential mechanisms whereby omega-3s can act. Membrane incorporation, which we somewhat rolled out. The production of oxylipids, which are simply biologically active oxygenated fatty acids, and you may not be familiar with the term oxylipin, but you've heard of prostaglandins and leukotrienes, which are examples of oxylipids. Activation of PPARs, which are nuclear receptors that function as transcription factors, and the newest mechanism is this activation of free fatty acid receptors. And there's some papers in the literature that suggest that the beneficial effects of omega-3s in inflammation and metabolism are mediated by FFAR4s. So we became interested in whether FFAR4 could mediate the protective effects of omega-3s. And the first question we had, since nothing was known about this, is FFAR4 or FFAR1 even expressed in the heart? And we found that FFAR4 is in fact expressed in cardiac myocytes and fibroblasts, but FFAR1 is not expressed, at least at appreciable levels, in the mouse heart. Furthermore, we were interested in if there was any baseline functional abnormalities caused by a deletion of FFAR4. So we're looking at systemic deletion of the knockout FFAR4, these FFAR4-KO knockout mice. We're going to use this as a model today. But we found at baseline they're pretty unremarkable. They don't really have a basal cardiac phenotype, and that's not all that surprising. Many cardiac phenotypes aren't uncovered until you actually stress the heart. So based on previous studies suggesting a putative protective role of the heart, we hypothesized that FFAR4 could function as a nutrient sensor for endogenous medium long-chain fatty acids to protect the heart from pathological stress. So that's what I'm going to show you today, how we tested that hypothesis. So the experiment, we're going to use FFAR4 knockout mice that I've already introduced to you to determine if FFAR4 is necessary for an adaptive response to pathologic pressure overload induced by transverse aortic constriction. And an important point I want to make, this is the first phase of a long-standing project we're going to examine, and these mice are only on a standard chow diet. We're not looking at any exogenous dietary fatty acid interventions. I want to be clear, there's no omega-3s being added to the diet in these experiments. And what I'd like to do now is take a brief moment to introduce the people who did the work. I've already mentioned that this study was done in collaboration with Greg Shearer's lab at Penn State, but these are the people who did the heavy lifting. So Katie Murphy, a postdoctoral fellow in my lab, Brian Harsh, a graduate fellow in Greg's lab, and Chastity Healy, my lab manager. So we did the experiment. We do the aortic constriction in the FFAR4 knockout mice, and then we looked at ventricular remodeling 4 weeks post-TAC, comparing the wild type in green to the KO in blue, sham versus TAC. And what we could see is that 4 weeks after TAC, there was an augmented hypertrophic response in the KO, shown here by an increase in heart weight and heart weight body weight ratio. We observed the same thing with myocyte cross-sectional areas, so we're actually looking at cardiac myocyte hypertrophy. We also noticed an interesting trend towards an increase in lung weights in the KO following TAC, with far more KOs showing this massive increase in lung weight, suggesting potential for a greater propensity to transition to failure in the KO. Now, interestingly, when we looked at fibrosis, that's shown here on the bottom left. This is picro serous red stain with fast green counter stain, and here's the summary data. We didn't observe any significant difference. When we looked at function 4 weeks post-TAC, we saw that in the wild types, again in the green, following TAC, there's a predictable decrease in ejection fraction that was further augmented in the KO following aortic constriction. When we looked at diastolic function by E to A ratio, we saw a small trend towards an increase in the wild type. It wasn't significant, but following aortic constriction in the knockout, there was a massive increase in the E to A ratio, suggesting diastolic dysfunction. There was also a notable trend towards an increase in end-systolic volume that related back to the decrease in ejection fraction we saw. So 4 weeks post-TAC in the knockout relative to the wild type, there was increased hypertrophic response with a notable trend towards an increase in lung weight suggesting the potential that they're transitioning at a greater degree towards failure. There was no significant difference in fibrosis and worse systolic and worse diastolic dysfunction. Now, another important point I want to make is we did observe a significant sex-based difference. The results I've shown you so far are only in the male mice. The female mice, the female KOs, adapted similar to the wild type. So the worst remodeling was only in the males. So from this, we would conclude that FFAR4 is required for an adaptive response to a pathological stress in the heart. But the question was, well, really, what is FFAR4 doing in the heart to protect the heart? And we focused on cardiac myocytes because we didn't see any difference in the fibrosis. And since nothing was known, we did a very broad-based experiment to get as much information as we could. We did a transcriptome analysis in cardiac myocytes from male wild type and knockout hearts, cardiac myocytes, 3 days post-TAC. So we perform TAC, we isolate the hearts, get the cardiac myocytes, get the RNA, and then do an RNA-seq experiment. And the first analysis we did was a principal component analysis to see if we could see any major difference in transcriptional responses in the wild type, again in green, to the KO in blue, sham versus TAC. And you can see surgery had a major effect on gene transcription that was bigger than the difference between the wild type and the KO. And maybe that's not surprising. But it doesn't really give you a lot of specific information about what might be going on. To do that, we did another analysis where we compared genes that are changed following TAC versus the sham in the wild type versus the KO that were increased or decreased 1.7-fold. And when we do that, we detect about 2,800 genes in the wild type in the big green circle, but only about 1,650 genes in the KO. And if we subtract out the common genes, about 1,400, we're left with 1,380 unique genes that were changed 1.7-fold upward down in wild type cardiac myocytes post-TAC, but only 247 in the KO. So quite a significant specific transcriptional difference. And to get a better handle on what those genes might be reflecting, we did an ontology sort for biological function that's shown here in panel C. On the left-hand side of the slide, here's the categories. We looked at genes that were specific to the wild type, specific to the KO, and those in common. And you can see the three ontologies had the biggest hits were cell death, inflammation, and reduction. And that's very interesting, and we're going to come back to that in a few minutes. But again, this doesn't give you a lot of specifics about what FFAR4 might be doing in cardiac myocytes. So to do that, to get a better handle on that, we turn to Ingenuity Pathway Analysis, which is a software program that's often used to analyze RNA-seq data. And using this, we created an FFAR4 signaling pathway in which we could overlay the gene expression based on prior knowledge. So let me walk you through this. So we knew from the literature that FFAR4 activates proximal signaling pathways, which would include beta-arrestin 2, G-alpha-Q, phospholipase E-beta, PKC, so on and so forth. So we added that to our map. From the work of Jared Oleski, we know that FFAR4 can activate inflammatory signaling pathways, and specifically activate beta-arrestin 2 to activate TAB1 and block TAC1, and then prevent this NF-kappa B pro-inflammatory signaling pathway. And in cardiac myocytes, this would be associated with inducing pathologic hypertrophy. Now, furthermore, we know from other studies in macrophages that FFAR4 can activate cytoplasmic phospholipase A2 to induce oxylipid production. That's shown here. Here's phospholipase A2. It cleaves out nuclear membrane fatty acids, and then those are acted upon by lipoxygenases, cyclooxygenases, P450 hydroxylases, and epoxygenases to produce oxylipids. And it includes genes like cyclooxygenase 2. Furthermore, we know from the work of Tom Forrest that if you knock out CPLA2, it increases IGF levels, and that can induce hypertrophy. And that's shown, that signaling pathway is shown on the left-hand side of the slide. And finally, from work in endothelial cells, we know that FFAR4 can again activate phospholipase A2 to increase expression of this gene called HMOX1, which you also know is heme oxygenase 1. This is a gene that reduces reactive oxygen species in the cell. It's known to be cytoprotective, and many experiments have shown that it is in fact cardioprotective. So when we overlay the gene expression data that we get in Ingenuity Pathway Analysis, it's upregulated in red, downregulated in green. We can see a major hit on these CPLA2 signaling pathways. Over here, looking at cyclooxygenase 2, cytoplasmic phospholipase A2, upregulation of this heme oxygenase response, and upregulation of beta-arrestin 2, which could theoretically inhibit this pathological response. Now, We don't see that in the KO. There's no upregulation of PLA2, no upregulation of HEMOX, and in fact, you see a couple of these very important P450 enzymes are actually downregulated post-TAC. So, based on this, we suggested that it suggested to us that FFAR4, CPLA2 signaling pathways, and cardiac myocytes are essential for an adaptive response post-TAC. And I'm going to focus on what FFAR4 signaling through CPLA2 and oxylipin synthesis does in cardiac myocytes for the rest of the talk. But since many of you are probably not familiar with this signaling pathway, I thought I'd give you a little bit background information. So when FFAR4 is activated, it can lead to activation of phospholipase A2, which then translocates to the nuclear membrane. And once there, it takes membrane phospholipids and cleaves them at the SN2 position to release fatty acids. Now, typically we think of that as arachidonic acid, but it can actually be any fatty acid that's in the position, including EPA and DHA. Once released, these fatty acids are further metabolized by cyclooxygenases, lipoxygenases, cyphydroxylases, and cyphaloxygenases to produce oxylipins. Now, once an oxylipin is produced, it can follow one of four fates. It can be resequestered into the plasma membrane. It can be left free in the cell, and in the cardiac myocyte, it can participate in a remodeling response. Or oxylipins can be exported, either as a free oxylipin, which really doesn't happen with any great frequency, or it can be exported in a lipoprotein vesicle. And in vivo, that would be in HDL, or high-density lipoproteins. And when we think about oxylipins, again, we're most familiar with arachidonic acid-derived oxylipins like prostaglandins and leukotrienes, and they're generally considered to be pro-inflammatory. But it turns out that EPA and DHA can also be metabolized by these enzymes to produce similar classes of oxylipins, and in general, these are considered to be anti-inflammatory. So our question was then, what is FFAR4 CPLA2 oxylipin signaling doing in cardiac myocytes? So the first thing we did was we cultured adult mouse cardiac myocytes, both wild type and knockout. We then treated them with the FFAR4 agonist TUG-891, and then after an hour, we collected the cells, collected the tissue culture medium, and we measured oxylipin production as a surrogate for CPLA2 activity using mass spectrometry. And we considered the four fates, either reesterified in the myocyte membrane, left free in the myocyte, esterified and exported from the cell, or unesterified and secreted as a free oxylipin in the medium, which again, really didn't happen with any frequency. Now, what we found was that when we looked at the knockouts, we didn't really see any response. The cells, they don't have the receptor, they don't respond to the agonist, that's not surprising. What we saw in the wild type was production of one specific oxylipin at the expense of the other 57 that we could detect. So quite a specific response. And that one oxylipin is 18-HEAP, also known as 18-hydroxy eicosapentaenoic acid. This is a pro-resolving oxylipin derived from EPA. And it's previously been shown that increased EPA levels, for instance in FAT1 transgenic mice, which overexpress an elongase to make endogenous omega-3 fatty acids, is associated with increased 18-HEAP in macrophages, and that's cardioprotective. Furthermore, 18-HEAP is known to be a precursor for E-resolvins, and resolvins, like other oxylipins, can act through a GPCR, and this GPCR is called ERV1 ChemR23, and that's also shown to be cardioprotective. The question remains, however, is whether there's any direct effect in cardiac myocytes. And to do that, we wanted to look at oxidative responses and cytoprotection in adult cardiac myocytes downstream in FFAR4. And as I promised, we're going to go back and we're going to look at that gene ontology sorting, looking at specifically oxidative reduction genes that were upregulated in the wild type that would suggest cytoprotection in the wild type post-TAC, but not in the KO. Genes including heme oxygenase 1 was upregulated 24-fold, cyclooxygenase upregulated 7.5-fold, glutathione peroxidase 7, which reduces sulfhydryl groups, was upregulated. Glutathione peroxidase 3, which reduces hydroxyl groups, was upregulated, as well as SOD3. So this would suggest that there is an adaptive response that is moderated by FFAR4 that is upregulated in the heart post-TAC that protects these cells, and it's mediated by FFAR4. But to really directly test that in cardiac myocytes, we cultured adult mouse cardiac myocytes, in this case just wild types, and we induce oxidative stress with hydrogen peroxide that's shown here, and not surprisingly, we've shown this many times, that this induces a cell death response. Now, when we pretreat the cells with TUG-891 overnight, this is again the FFAR4 agonist, it can attenuate that hydrogen peroxide-induced cell death. So we see FFAR4 in vivo induces cytoprotection by upregulation of genes that could reduce oxidative stress post-TAC, and TUG891 specifically prevents cell death from oxidative stress in wild-type cardiac myocytes. Now, mechanistically, we were interested in what role 18 heat might play. So we did the same experiment, in this case, in wild-type and knockout cardiac myocytes. So in both cases, we can see that hydrogen peroxide increases cell death. That's shown here in the summary data. Here's the increase in cell death in the wild-type and in the KO. If we pretreat the KO, wild-type and KO cells with 18 heat for 2 hours prior to hydrogen peroxide, you can see it attenuates hydrogen peroxide-induced cell death. So 18 heat prevents cell death from oxidative stress in both the wild-type and the KO, suggesting a mechanistic link between activation of FFAR4, production of 18 heat, and cytoprotection in cardiac myocytes. And it's this cytoprotective mechanism that could explain some of the beneficial effects of FFAR4 in the heart. The absence of this signaling pathway could contribute to the worst pathological remodeling seen in the knockout following TAC. Now, going back to the data I showed you a few slides ago, one of the things that we found was that 18 heat was also exported from the cell and lipoproteins. And in vivo, the way that works is when these oxylipids are produced intracellularly, they are exported and trafficked in plasma high-density lipoproteins. And HDL, the largest reservoir for plasma oxylipids, and HDL directly acquire their lipids from peripheral cells. So 4 weeks following TAC, we isolated plasma lipoproteins, specifically HDL, from the wild type and the KO, again in green and blue, and compared sham to TAC. And the first thing we did, again, like the gene expression data, was to do a principal component analysis to see if we could see any global differences. And in fact, we saw some significant differences. When we looked at the wild type sham animals, their HDL were primarily consisting of anti-inflammatory oxylipids, for instance, 13-HOD. Whereas following aortic constriction, there's a shift in the oxylipin content. We see an increase in the pro-inflammatory arachidonic acid oxylipin called 15-HEAP, as well as an increase in the EPA-derived pro-resolving oxylipin 18-HEAP post-TAC. So there's an adaptive response. Let's compare that to the KO in the blue, the shams in the open circles. They're largely dictated, their population is dictated by 9-HEAT. What is 9-HEAT? It is a non-enzymatically derived oxylipin. It's a product of auto-oxidation. And that dominates what you see in the KO sham. There's a little adaptation post-TAC, but really nothing like we see in the wild type. And if we specifically consider 18-HEAT, the oxylipin I showed you produced in cardiac myocytes downstream of FFAR4, if we look in the wild type animals, in the sham condition that's represented here, they have a fair amount of 18-HEAT that's increased post-TAC, and that's shown here. What you see here in the KOs, they don't really have any in the baseline, and there's a little bit of an increase post-TAC, probably from other mechanisms that can produce 18-HEAT, but it never even reaches the baseline level that you see in the wild type. So systemically, FFAR4 maintains anti-inflammatory oxylipins at baseline and increases their inflammatory and resolving oxylipins in HDL post-TAC. So the final question we wanted to address is whether there's any role for FFAR4 in the human heart. So to do this, we first wanted to measure FFAR4 and FFAR1 expression in the human heart, and that we did in collaboration with our friend down at University of North Carolina, Brian Jensen. And Brian's lab measured FFAR4 and FFAR1 expression in non-failing and failing hearts. So first, let's look at FFAR4. You can see expression of both the short and the long isoform. Again, the predominant isoform is the short isoform, about 25-fold more than the long isoform. And interestingly, it was downregulated by about 2-fold in failing hearts. Compare this, though, interestingly, we found FFAR1 is expressed in the baseline non-failing hearts. Now, we did not see expression of FFAR1 in the mouse heart, so this is somewhat new biology, and even more interesting, it's increased in failing hearts by about 2-fold, and whether this represents a compensatory response, we don't know, but we did get a little bit nervous that maybe we previously missed the detection of FFAR1. So we went back and we reanalyzed FFAR4 and FFAR1 expression in the mouse hearts, wild type in the KO, and you can see we got what we previously got. It's got FFAR4, not FFAR1, and there's no upregulation of FFAR1 in the KO. So none of this type of compensation we see in human hearts. And this is some new biology, so we're going to have to figure out a way to pursue this. since FFAR1 is not in the mouse heart. Well, the next question we had is there any functional relevance of FFAR4 in the human heart? And to address this, we turned to our friend Quinn Wells at Vanderbilt University, and what Quinn did was an ECHO-WAS study. Now, that's kind of like a phenome-wide association study, except for you're using echocardiographic data. So Quinn pulled electronic health record data from patients who were referred for echocardiography at Vanderbilt. And then we looked at known polymorphisms of FFAR4 and tried to determine if that had any effect on cardiovascular function. And the one FFAR4 polymorphism that we focused on was this R270H polymorphism that was previously identified by Gozo Tushimoto and Phil Frogel's group in a paper in Nature in 2012. And in that paper, they showed that this polymorphism was associated with morbid obesity in humans, BMIs of greater than 40. They also showed that this polymorphism is located in the third intracellular loop, and in signaling experiments, they showed it's a signaling dead mutation. In other words, it can bind the fatty acid, it just can't activate downstream signaling pathways. So in a sense, it's kind of like having a knockout receptor. So what Quinn did was looked at the electronic health record data for the echocardiography. This R270H mutation polymorphism was found with a minor allele frequency of about 1.8% in the roughly 7,000 cases that were analyzed. And what Quinn found was that there was a significant increase in LV mass, ventricular dilation, and left atrial diameter, as well as a notable trend towards a reduction in ejection fraction. So in the human heart, this R270H polymorphism appeared to be associated with hypertrophy and ventricular dilation, very much like we saw in knockout animals post-TAC, where we saw worse pathological remodeling. So in summary, what have I shown you today? That first of all, FFAR4 is necessary for an adaptive response to pathological stress in the heart. And this cardioprotection occurred independent of dietary intervention, implying that FFAR4 senses and responds to endogenous fatty acids to protect the heart. Now, we don't know at this point in time which fatty acid or fatty acids may be having this protective effect. And furthermore, we want to go on further to test whether FFAR4 is required for omega-3 cardioprotection. We just haven't had time to do that yet. Interestingly, in cardiac myocytes, we found that FFAR4 specifically and uniquely produced the EPA-derived cardioprotective and pro-resolving oxylipin 18-hep. Now, that's interesting for a couple of reasons. It's just about the only example of a receptor-mediated production of oxylipin that shows this type of specificity, one oxylipin. Not only that, when you consider that EPA is scarce in cardiac myocytes, it represents only about 0.5% of the total membrane fatty acids that you see in a cardiac myocyte. For comparison, DHA and arachidonic acid each account for about 10 to 12% of the total fatty acids. So this is really remarkably specific. Mechanistically, we saw that FFAR4 protected wild-type cardiac myocytes from oxidative stress-induced cell death, and 18-hep protected both the wild-type and KO, suggesting a mechanistic link between FFAR4, production of 18-hep, and cytoprotection. And the absence of this protective signaling pathway may have contributed to the worse remodeling we saw in the KO post-TAC. Systemically, the loss of FFAR4 was associated with less EPA-derived anti-inflammatory oxylipins at baseline, and inability to increase pro-inflammatory and EPA-derived pro-resolving oxylipins, particularly in HDL, following TAC. And finally, in humans, we saw that FFAR4 expression was evident and decreased in heart failure, while the signaling deficient FFAR4 R270H polymorphism was correlated with hypertrophy and ventricular dilation in a large clinical cohort. Now I'm just about done, but again, want to really point out the people who played a major role in this project. Again, all this work was done in collaboration with Greg Shearer's lab at Penn State. And Greg and I have been working on this project for the last 12 or 13 years. But the people who really did the heavy lifting, Katie Murphy, a postdoctoral fellow in my lab, Brian Harsh, a graduate fellow in Greg's lab, and Chastity Healy, my lab manager. I also want to give special thanks to Brian Jensen at the University of North Carolina for measuring FFAR4 and FFAR1 expression in human hearts. Nathan Tintle at Dordt University, he's our statistical guy. He does all our statistical analysis and reviews everything for us. Quinn Wells at Vanderbilt University for his work on the ECHO-WAS study, and I want to thank our current funding sources from the NIH and prior funding for this project from the American Heart Association and Amarin Pharmaceuticals. Thanks very much. And I'm going to get out of this. Let's see, stop share.
Okay, thank you very
Okay,
much.
we're
Thank
back.
you very much, Tim.
Yeah,
Very excellent
absolutely.
talk. I forgot to mention that if people could please put their questions in the question and answer box, and Tim's going to go through this, but I'll take my prerogative as the chair to ask the first question. And I was
Yeah.
wondering, in your FFAR4 knockouts, what kind of profile of fatty acids is there a delivery change of the type of fatty acids that the heart sees?
It's an interesting question. So we measured total fatty acids, everything from C10 up to C24. Didn't see a whole heck of a lot of difference in heart membrane fatty acid content in terms of that kind of a global look. So I can't really say that we saw anything. There's really no baseline difference. Now, the one thing is we looked post-TAC, and again, we didn't really see anything majorly different in the total fatty acid content. So, one of the open questions that we would have then is, what the hell is the ligand for FFAR4? How is this stuff working? It's a big open question, and really, most of the studies have been done in vitro, which really you don't replicate what you're seeing in vivo, in my opinion.
Mhm.
And so it's a very difficult question to understand, and there's so much about this receptor that we just don't know. So, but that's a great question because we thought about that. That's exactly what we were thinking.
Oh, great. So I'll let you go to the question and answer and go through them.
Okay.
We have quite a number now, and they're coming in. So go ahead and.
Okay, so the first question is from Dave Bearfield. The variant you linked to in the ECHO-WAS study was fairly high frequency. Were there any demographic data included with the ECHO-WAS analysis? Did the variant associate with any specific populations? That's a good question. One thing I can tell you is that we did look at African American patients. We did look at white patients. We just didn't have enough data. We didn't have enough sample size in the African American patients to make any meaningful conclusions. Further than that, I can't give you any more specific information. They were all from Vanderbilt, so I guess in the Tennessee area, however homogenous that might be, obviously we'd want to look at other patient populations to see if this still holds true. The second question from Sam Dudley. Hi, Sam. Sam is the director of the Lehigh Heart Institute here at University of Minnesota. So, Sam, I hope you're doing well. If I remember correctly, PUFAs are substrates for ALOX that produces resolvins, anti-inflammatory molecules that participate in an active process of resolution of inflammation. Could this be how PUFAs work? That's exactly one of the things that we've been thinking, Sam. So resolvins, particularly the E-class resolvins, are, and I mentioned them in the talk, they're derived from EPA, but there's also D-class resolvins that are made from DHA. Now, one of the interesting things that we saw in this is that FFAR4 can specifically activate production of 18-HEAP. So that is the precursor for E-resolvins. So I do think it's very interesting that EPA, that could function as a receptor ligand for FFAR4 and then produce 18-HEAP, which can then produce E-resolvins, which are known to be cardioprotective. And there was a very nice paper in circulation about, I'm going to say it was either 2018 or 2019, looking at the receptor for resolvins called ERB-ChemR21 in atherosclerosis, and showed that it was very important for attenuating atherosclerosis downstream of EPA supplementation. So yeah, I think that's right on track, some of the things that we're thinking about. That's one potential mechanism. Are there body weight differences in the KO in males or females or both? This is from an anonymous attendee, so I can't tell you who that was. Is anything known about blood glucose in the FFAR4 KOs? Yeah, there's a whole literature on this in terms of, and it's somewhat confusing because it's conflicting. Some people have reported that at baseline there's not a lot of difference. There may be decreased insulin sensitivity in the KO, but when you feed them high-fat diet, the KOs in some people's hands get, if I remember correctly, to think about this, I think they get fatter, but some people have not been able to reproduce that. So there's been a little bit of a debate about that. So, anonymous attendee. Am I reading this right? 4 weeks after TAC, your KO versus wild type, there's a significant difference in ejection fraction, but there is no difference between the wild. Yes, that is exactly correct. You're reading that correctly. Xavier Ravello. Hey, Xavier. Xavier is right down the hall from me. He's one of our new faculty members here at University of Minnesota. So nice to see that you're doing well, or at least well enough to text in. Xavier, could you speculate on what might the endogenous FFAR4 ligands that would mediate the cardioprotective effects, considering your mice were on a normal child diet? Yeah, that's a great question. I wish I knew, because we're talking about free fatty acids. And when we think about cells, most of the fatty acids are not free. So we're going to have to figure out some really elegant ways to look at what fatty acids actually might be free in a cardiac myocyte in vivo. Because one of the challenges we have with cardiac myocytes in culture is, could we actually replicate fatty acid content in vitro versus in vivo? And that's been a big challenge for a lot of studies that are done in cells.
Sorry, Tim, just to interject. In the previous question by the anonymous attendee, I think they were actually asking for some resolution on why there isn't a change in ejection fraction following TAC, even in the wild type, I suspect. So
Okay, all right. So, well, in our hands, what we're seeing is we specifically looked at a 4-week time point, which is a pretty early time point when you're looking at TAC. So you could see, if you looked at the spread of the data, there were some animals that were transitioning more than others in the wild type, but in the KO, they had all transitioned down.
Sure.
And that's where the significant difference came. And so it's not that there was no decrease in any animal in the wild type. That's why, if we go back to David Eisner's talk, that's why we plot these individual data points. And there's a great variability, specifically when you look at this 4-week time point. And to go back to originally why we looked at the 4-week time point, when we submitted the grant to NIH and we wanted to look at this was one of the specific points that all the reviewers pointed out. They're like, no, you need to look at this early on, specifically if you're thinking there could be any effects on diastolic dysfunction, diastolic remodeling, which is originally where we were thinking this project was going. So, yeah, okay, I hope that is a better answer.
I think that makes sense.
Okay. Sam asked, since I think this was answered, yeah, okay. Was there an expected reduction in NLRP3 inflammasome or mitochondrial oxidative stress? We didn't look specifically at that, Sam. We just haven't gotten that far. The next question is from Thomas Polincanal. I'm sorry if I mispronounced your name. Nice talk. Could you give a comment on cardiac triglyceride content in the KO mice? We didn't look at the triglycerides specifically, so I can't address that question. It's something actually that Greg and I are talking about doing going forward and looking at function specifically in the adipose. We just haven't gotten that far yet, but it's a good idea. We want to do that. Emiliano Medei. I hope I said that right. I'm sorry if I didn't. Great talk. What about the systemic and local inflammatory profile? It's a great question. We're working on that. Can't give you anything yet. Sorry, I can't say more, but we're shut down. That was what we were working on when we were shut down, so we haven't gotten that done yet. Since female KO mice show no response, have you carried out the in vitro analysis in cardiac myocytes from female KO animals, or compared the gene expression post-TAC in females? Yeah, we haven't done the gene expression in the post-TAC in the females. And, it's something that we just got comments back on a grant where we were trying to extend this project further, and they wanted to know more about what was going on in the females. And we just really haven't done a lot with that. We only have so much bandwidth in the lab, and while that's a great question, we just haven't had a chance to tackle it. Jason Karsh, this is the question that we're trying to do right now. Do omega-3s display a protective effect against TAC on the FFAR4 KO mice? Well, we predict that if FFAR4 is required for these cardioprotective effects, no. We haven't done the experiment yet. Again, bandwidth. We only have, I don't have a huge lab. I don't have 40 people in my lab. We can't answer all these. And these experiments take months and months and months to do with all the analyses that go into them. What are the future directions in this line of research? From Kaha Shamim Mohammed. That's an excellent question. We're now looking at a bunch of things. One of the things that we now know is molecularly, how this receptor actually works, what ligands it sees. And to reference a question that came up earlier, we're very much interested in inflammatory responses in the heart, and we're starting a new project looking at FFAR4 regulation of inflammatory responses post-MI. So we just got a new grant to study that. So those are some of the future directions. And anonymous attendee, have you looked at the effect of FFAR4 on mitochondrial respiration in adult hearts? No, we haven't, but Julia Liu, I'm gonna put a plug in for her. She's a new faculty member in our department. She came from Tisha Murphy's lab at NIH. She's a mitochondrial expert, and we may be walking down the hall to talk to Julia Liu about some of this, because that's an excellent question. Casey Wright, a former postdoc in my lab. Great stuff, Tim. Thanks, nice to hear from you, Casey. I hope you're doing well. Shan Mugasandaram Paki, I can't see the whole last name, it's cut off. I'm sorry if I mispronounced that. Thanks for the talk. Can you comment on fatty acid binding proteins in relation to FFAR4 signaling? Yeah, so this is something that Dave Bernlohr, who is our chair of biochemistry, Dave is an expert on fatty acid binding proteins. And Dave and I have been threatening to start a project on this, but again, we're on lockdown now, so we haven't gotten that far. Ira Goldberg, nice to hear from you, Dr. Goldberg. In a very large Resolve It trial, the EPA benefit was vascular, not changed in heart failure. That is correct. Is there a subgroup that you might ask Aaron to look at? Yeah, we've tried. We haven't gotten any look at it yet. So, and I'm not sure what that subgroup would be. We haven't gotten access to the full data set, and Greg and I are kind of anxious to get that. So we'd really like to get that. But thanks for your question, Ira. Wow, this just goes on and on. But that's great. That's no problem. Adrian Arietta. Given the antioxidative effects of FFAR4 signaling and its linked to fatty acid metabolism, could you comment as to whether FFAR4 could affect atherosclerotic plaque formation? Yes, in fact, I think it does affect atherosclerotic plaque formation. It hasn't directly been tested, but I'll put a plug in for another one of our young faculty members here. Jesse Williams studies inflammatory responses in atherosclerotic plaques. Jesse trained with Gwen Randolph at Wash U, and so he's an expert on this, and I would love to do a project on that with Jesse, but as far as what's been looked at in the literature, it hasn't been specifically addressed. Emma Robinson, thank you a lot for your talk, and I will definitely now catch up with your paper. Oh, well, thank you. Have you had the chance to look at FFAR4 in response to physiological stresses where the response is deemed predominantly adaptive? I'm presuming you're thinking about exercise responses. That's a good question. We haven't even thought in that vein yet, but that would be an interesting question to answer, particularly if we challenge the mice with some type of high-fat diet or a metabolic syndrome model, kind of like akin to what Joe Hill is working on. What would these mice do in that capacity in terms of activating FFAR4? And that's something actually we're working on in the lab right now. Jitka Virag, great work. Any ideas what happens to FABP binding levels? Yeah, no, somebody asked that earlier. We haven't gotten that far. It's something that Dave Bernlohr and I have talked about a little bit, starting a project in cardiac myocytes, but we really haven't gotten anything up and going yet. And finally, Paul Simpson, hey, Chief, how you doing? It's good. I hope you're doing well. Can you say something about external agonists? What is TUG? Yeah, so there's a fair amount of pharmacological work on designing agonists for FFAR4. One of them, the most prominent one, the one most commonly known is TUG 891. However, I will say that most of the experiments with these types of drugs have only been done in vitro, in cell culture. I did a lit search because we were interested in actually using TUG 891 in like an in vivo model to see if activating FFAR4 was protective. And there's exactly 2 papers that have tested it. And they used wildly different routes of administration and different doses. So yeah, I don't know. There is a whole literature, and Graham Milligan, who I think you know, Paul, is leading the charge in this. He's done a lot of work on FFAR4 agonists and specifically looking at targeting receptor and receptor signaling mediated pathways. But again, most of it's been done in vitro. There's almost nothing done in vivo at this point in time.
Tim, you could tell us what BG stands for? Is that big guy? Is that
Big
your
guy. Yeah.
nickname?
He's always called me big guy since I've been in the lab for the last 25 years. And yeah, he's just, if you haven't met Paul, I suggest you all take the chance to meet Paul. He's one of the greatest people you'll ever meet and probably the smartest guy I know. Sharinda Rudiger, another friend of mine who's in Germany right now, if I'm not mistaken. I think she's still in Germany. Hi, Tim. Nice study. You're so fast. Sorry if I missed it. What did you say could link FFAR4 activation to the OXRED gene induction, oxidative reduction gene induction? Yeah, we don't have specific signaling pathways linked to activation of gene transcription yet. One of the things, working with primary cardiac myocytes and studying signaling pathways, although it's something I've been doing for like 20 years, it's not the easiest thing to do. And we just haven't got it. Not for lack of trying. We're working on it, but we just haven't gotten that far. So thanks. Bruce Allen, great work, Tim. Nice to see you online, Bruce, lurking in the back, as you said in your email to me. Yes, still in Germany. Casey Wright, agreed. Paul's the best, from Casey. Okay, so that brings us to the end, and thank you everybody for attending. Thanks again to Devor. Excellent job, mate, on wrangling this whole thing. I can't believe the amount of work you've put in on this. And Gary, thanks again for introduction and moderation.
Well, thanks a lot, Tim. It's a great talk and quite a number of questions. 23 in total, although 4 or 5 of them were comments. And congratulations. Yeah. It was a great talk. And with that, I'll pass it back to Devor, I guess, and we'll look forward to future seminars.
Okay.
Well, I'd like to thank you, Tim. Really, really great work, great science. Thank you for actually showing all the data that's in the process of getting published. It's really nice to hear these stories before we can read about them, actually. So it's really great, and thank you for allowing it to be recorded as well. Thank you for answering lots and lots of questions, and Gary, of course, thank you for volunteering to chair and for actually chairing in such a nice, relaxed fashion as well. Audience, thank you for tuning in, and we will see you tomorrow. And hope you have a nice rest of the day, or good night, whichever way, wherever you are.
Have a good day.
Have a great day. Thanks, everybody. Bye.